back to Science with Pierce. I'm your host, Pierce. Thank you once again, and as always, for joining me. Today, I'm going to discuss a revelation I've had over the past month or so, which I think could be best described as the fact that the boundary between food and matter is nothing more than a convention. And any convention can be transcended if we can first only but conceive of doing so. And so, well, I'll just roll the footage. All right, so I know what you're probably thinking. Maybe you're saying to yourself right now, Pierce, why the f*** did you make human dog food in that video? And why did I just watch it? And it's a fair question. You know what? Yeah, that's exactly what I did. And I did it for science. So the experimental design is pretty simple. I measure some baseline things about myself, like my weight, blood pressure, how much sleep I'm getting, psychological, sort of subjective measures of psychological health, and several other things. And then I eat this food and only this food 3 times a day for every single day for an entire month. And throughout that month and after the month, I measure those same things. But more importantly, though, I'm measuring another, kind of a focal output, which I think is the most interesting readout for the whole experiment, which is my gut microbiome changes in at least the diversity and the absence of gut microbes. And I'm using a kit from uBiome or Microbiome, a company based out of California that uses a technique called 16S ribosomal RNA sequencing to measure the microbes inside your gut. And this is because it's increasingly becoming known and popularized that the microbes inside of your gut play a pivotal role in not only your health, physically, but also your psychological health and several other important things. So I wanted to understand how eating a single type of food was going to affect my gut health. And I wanted to make sure it was a complete food. And a lot of people will tell you that the way to have an unhealthy gut microbiome is to make sure that you eat foods that are not diverse and foods that are not varied. The less variable your diet is, the more unhealthy your gut microbiome will be. And I think that's probably true. But that's not what I wasn't trying to make a healthy gut microbiome. I was trying to understand, with one type of food, what is the result? And that's what I hoped to have discovered in this experiment. So, I mean, before I go into why I chose each of the ingredients and so the results from the experiment, I guess I just want to first describe like what the experience of eating the food was like. So, I can tell you that it didn't taste bad at all. I mean, it wasn't amazing. I mean, it has like a sort of a subtle sweetness to it, a little bit of coconut in there for the fiber. First time in my life I've gotten this much fiber. Holy crap! But the overriding flavor is of oat and oatmeal. And that's because the proportion of the food, which is oat flour, is large, because that's where the primary source of carbohydrates is. And if I could make it again, I probably would change a few things. Like the moisture content wasn't quite right, and I used emulsifiers and binding agents like soy lecithin and xanthan gum, and I think that was a correct thing to do. But I think that the amount of each of those should be changed slightly, because it kind of left it with an almost rubbery kind of feeling. So yeah, it wasn't perfect. If I had to describe it though, in terms of the texture, I'd say it's kind of like eating compressed, dense cornbread, kind of like, but like also sort of like eating the inside of some like power bars. I can't quite explain it, but needless to say, if I plan on making a startup around this product, I have a long ways to go. So yeah, now we'll go into why I chose each of the ingredients. All right, so if you were interested in my reasoning behind each ingredient, this next section explains that. We'll start with protein. I included soy, whey, milk, and algae protein in this mixture because I wanted to create an amino acid profile which was diverse, and so that I'm not at risk for a deficiency in any one amino acid, but also because these different protein sources have amino acids in different configurations, right? So soy and whey tend to have more BCAAs, or branched chain amino acids. And BCAAs are great for people who lift weights because they're considered to be in a form which can be easily and quickly used by your muscles. But the problem is I don't want to have just an immediate and sudden burst of amino acids after I eat a meal, leaving me with no protein for the next 5 hours between each meal. So as a result, I chose milk powder, which is rich in casein, which would serve as my source of slow protein. Casein is slower to digest due to its poor solubility and high molecular weight compared to BCAAs. But this way I'm hoping to deliver a steady stream of amino acids into my body over the course of the day so that my muscles will never feel like they have a need to digest themselves, and instead remain in a constant state of anabolism, that is to say, muscle building. So I chose algae in the form of spirulina because it's got an incredibly high protein content. It's got more vitamin A and beta carotene than anyone could really ever need. It's got choline, polyunsaturated fatty acids, omega-3s, omega-6s, trace minerals, all this stuff. And actually, the only reason I'm not surviving off of 100% spirulina is because it tastes terrible, and also because I don't want vitamin A toxicity, which you can get if you eat too much spirulina, but it's not a huge issue. And then, like I was saying, the main source of carbohydrates besides the carbs in the milk powder is the kilogram of oat flour I'm to consume each week. I didn't cook the oats because cooking oats actually reduces their nutritive potential. And oats are high in fiber and protein, minerals, and vitamins. But some people think that oats are high in phytosterols and phytoestrogens. And that is something which they think will cause unwanted side effects, hormone imbalances. But I've seen little to no evidence to support that phytoestrogen intake can cause endocrine disruption. So I'm not convinced about that. But we'll see. The reason for the xanthan gum and the soy lecithin is because I want this food to stay together as a solid. Xanthan gum contributes to my dietary fiber content, but really these two ingredients contribute mostly towards the texture. Soy lecithin is an emulsifier, and xanthan gum is a stabilizer. It keeps things together, and they're both extremely benign in terms of health effects, so really not much to say there. Coconut flakes and chia seeds, they're there for the fiber mostly, and also the texture and the flavor, but mostly fiber. I mean, one of the main things in this diet is changing about my normal food intake is that for the first time, maybe in my entire life, I'm going to be getting the recommended amount of fiber every day. And I expect this will change a lot about my gastrointestinal comfort, but it will also provide a food source for the microbes in my gut so that they can flourish more easily. Because these fiber-rich ingredients aren't meant to feed me. My body can't digest these complex carbohydrates, but the bacteria and fungi inside of my gut
can digest it. So it's there for them. Let's see what else. Oh yeah, the cashews are there for the fat, the protein, carbs, everything. Cashews are expensive, and I don't really like to use them because of the fact that cashews are not an extremely land efficient crop in terms of biomass per acre. Well, I guess I should say biomass output per acre, but man, they're so darn good, and they have so much nutrition. It's nuts. Get it? Nuts? Okay. The salt is important. It's actually probably one of the most important parts. It won't taste like it's supposed to taste if you don't put salt in it. That's true of almost anything. And sodium is essential to survive, but the really important thing about the salt is that it's iodized. And in order to have proper thyroid function, you need to get enough iodine. And so the way that humans normally get enough iodine is through consuming foods which contain iodized salt. And so I was very careful to make sure that the salt I was adding to my food was indeed iodized so that I could have proper thyroid function. I actually added extra B vitamins here because I have gotten my genes probed by a microarray, and according to the analysis of the data, I require slightly more B vitamins than average to remain healthy. So I included vitamin C as well. There's no citrus in my mixture here, and vitamin K because while my food is green, it's not quite a vegetable. You're probably wondering about the wheat germ oil, and that is there because it provides additional polyunsaturated fatty acids, but mostly because it provides all the vitamin E I will need to maintain good health. And it tastes terrible. I mean, really bad. It smells bad, tastes bad. And I was really glad to find that the flavor didn't come through in the finished product. And so, that was really, really exciting. The vitamin D is the fat-soluble vitamin I added to the wheat germ oil as well. And that's there to make sure I don't get osteomalacia and also so that I don't get depression. Okay, so here's the actual nutrition label for my food. And we'll see if this works out.
Okay, so let's talk about the results of my experiment, and I'm going to show you some slides that I prepared. Okay, so we'll start with blood pressure. Pretty flat. I don't think anything's wrong here. Pretty stable and normal for a young man. With weight, I saw a slight increase, but hardly any. So I went from average of around 176 to about 179. This is 75 kilograms to about 78 kilograms. So actually that's pretty significant now that I think about it. Kilograms weigh a lot compared to pounds, but I think that this is marginal and actually could mainly be due to the fact that I was getting 3 meals a day, whereas usually sometimes I work through meals instead of eating or I forget to eat. So this is this could be a real trend. I'm not exactly sure though. In terms of sleep, I get more sleep on the weekends and I get very little sleep during the week. And that is pretty much all that indicates. Sometimes I sleep way too much, as you can see. But that's my right, and I'm entitled to it. Heart rate, around 60 to 70, pretty flat. I don't think anything's changed there. And then we have stool consistency, and this is based off of the Bristol stool scale, where 1 is, more difficult to excrete, and 6 is, stool which wants to excrete on its own. And you can see that in the beginning I have pretty inconsistent stool because I have an unhealthy gut microbiome, and then about 2 weeks into the experiment it becomes even crazier. And then it becomes very consistent, 3s and 4s, 3s and 4s, 3s and 4s. And so, this period of chaos may have been worth it for what the outcome was. But it seems that this kind of maybe provides a time horizon or a time frame for which serious improvements in gut microbiome health might take place. And so for me, it was like 3 weeks. And then I wanted to measure some psychological outputs, and these are just based on my own evaluation of myself, so maybe not super scientific, but for this one, agreeable/hostile, this is based on how agreeable I think I am. So higher agreeability is higher on the y-axis, and I think that if you would have my friends judge me, they would say, yeah, this is accurate. I'm not known for being an agreeable person, and this is pretty consistent as well. Here we're looking at composure versus anxiety. Composure is associated with a number on the y-axis. I'm a graduate student, so I live in a constant state of fear. Clear-headed/confusion. Again, I'm a graduate student. I go through, very cyclic bouts of feeling like I know nothing and then feeling like I know everything. Mostly nothing, though. And that goes right along with this next one, which is confident versus insecure. So I have like moments where I feel very confident and then moments where I don't at all. And yeah, for some reason, though, I keep coming back to the confidence before the cold hand of reality slaps me back into my place. But, yeah. And then I have energetic versus tired, which I think a lot of people are interested in because, a lot of studies have suggested that your energy level can change, or your energy level can be changed by the gut microbiome composition. But for me, I didn't see a huge change in my energy levels over the course of the entire experiment. And if you're wondering why I'm measuring all of these psychological outputs, it's because there's, a lot of really, really good evidence to suggest that people can predict your psychological health based on just nothing more than a sample of your stool. They found, they were able to predict whether someone had schizophrenia based only on their stool with something like 95% accuracy. So, I think that I'm very well justified in looking at these subjective measures of psychological health, even if they are crudely measured, and just because it's possible that what you eat can change how you feel in your brain. And so that's why I studied that. But back to the data, we look at the actual metagenomics here, look at the microbial changes. Probably the biggest change I saw was in the ratio between Bacteroidetes and firmicutes. So this is the before. This is before I actually started my diet. I took a sample of my stool and sent it off, and you can see that the firmicutes are exceeding, in terms of number, the Bacteroidetes. Overproliferation of firmicutes has been associated with diabetes and obesity, and general bad health. And so it was interesting for me when I saw that there was a reduction in the number of firmicutes in the during sample, and then a stark reduction in the after sample. And so I can see now that I have a higher proportion, or I did, maybe I don't now, but I did have a higher proportion of Bacteroidetes than I did firmicutes at the end of the experiment, which indicates to me that something significant changed here. And then I'll show you guys a few more really interesting things I found. One of them was this Akkermansia improvement. So before, during, I had about average compared. So they compare your proliferation or your abundance of Akkermansia relative to other people, and then they get an average, and they say, okay, you're at the average, or you're a little below the average. So the during, I had a decrease in the during phase, but then somewhere between the during and after sample, there was an explosion of Akkermansia, I guess. And I don't know what that was about, but I find it very interesting because Akkermansia is also associated with reduction in risk for heart disease and diabetes, I think. And so, I think that's quite interesting. I also saw an increase in the bifidobacteria content of my gut, and that's interesting because, bifidos as well as lactobacilli, which I did not really see a big change in. Those are both considered probiotics. And, I did have lactose in my gut because of the dry milk powder. But I don't know if, that's really a significant change. But what I can say is that based on the Akkermansia change in proliferation and also the change in the ratio of Bacteroidetes to firmicutes, I can pretty safely say that there's positive improvement in terms of like my risk for heart disease and diabetes. Not that I was really that much at risk in the beginning, but this was a positive change in my health, and I measured it and induced it using this method. And so overall, one of the things I found interesting was that, the relative prominence of each of the bacteria that was measured in the test didn't change that much in any, discernible pattern other than the ones I mentioned. And that could be because before, during, and after, on the Simpson's Diversity Index, which is an index that measures the diversity of the microbes in your gut, I rated like an 8 or a 9, which is actually above average. But I would say that some of the other interesting things that happened to me were that I was eating more than I ever have before in terms of calories, but I was getting hungrier faster after each meal. So I would eat a meal and then it would not sate me for very long. So I was going between each meal like a long time, feeling very hungry. And so that was interesting because I was getting, calorically, I was eating more than I usually ever do. So that was strange. And then, there was a really overpowering and lingering craving for fried chicken, which was prevalent, the entire time. Which makes me sad because I wanted for this to be something that could sate me and I could live off of like a substrate and, not have cravings for fried chicken. But what was also interesting was that, I thought the cravings would go away after a little while, but they just became stronger. So whether this indicates that I was not getting something that I needed, or if I just really like fried chicken, I don't know. So I hope you'll join me again on this channel where I do fun science things and fun computer nerd things, all sorts of stuff. I hope you'll subscribe and give me a thumbs up and share this with your friends if you think they might like it. So